Hi everyone, my name is Jonathon Stone. My title of my thesis is the development and validation of a 3D printed dosimetry phantom for Paediatric Radiology. And my supervisors for this project were Rick from MTP at Sir Charles Gairdner Hospital and Pejman from UWA. So first of all, just an introduction. So anthropomorphic dosimetry phantoms have several applications in diagnostic radiology, including radiation dose measurements to organs, especially for complex imaging modalities such as DXA, dual energy CT, and cone beam CT, as well as the optimization and justification of these radiology procedures. We can also use anthropomorphic dosimetry phantoms for obtaining exposure factors under automatic exposure control, and we can also simultaneously measure image quality parameters. So there are two types of anthropomorphic phantoms. First are physical phantoms, like the rando phantom here. So they're human-like in appearance, and they provide an accurate representation of human anatomy. And quite often their tissue compositions are based on international standards like the ICRP. And these phantoms also facilitate dosimeters for dosimetric measurement. The second type are computational phantoms, like the MIRD and ORNL Oak Ridge National Laboratory phantom series. And so these are built from simple geometric volumes using mathematical equations. And quite often the tissue compositions are based on the Christian and Eckermann 1987 phantom composition. So these phantoms are quite often used in Monte Carlo simulation software because of their simple geometry. And some of these Monte Carlo simulation software for radiology include P6MC, CT Expo, Radometrics, and Aerodex. So P6MC 2.0 or PC program for X-ray Monte Carlo is dose simulation software for planar radiography, and it's used by the medical physics team at Sir Charles Gairdner Hospital and Perth Children's Hospital. So P6MC allows you to cover a wide range of patient sizes, and you can also adjust many different beam parameters such as KEV, MAS, field size, and filtration. So the P6MC phantom uses the Christie and Eckermann 1987 phantom, which is one of the most commonly used ORNL anatomical phantoms. And this phantom is built by several parametric equations, which are used to build geometric volumes. So the phantom consists of 5 homogeneous tissue types, which are soft tissue for newborn and adult configuration, bone tissue for newborn and adult configuration, and lung, which is the same for both the newborn and the adult. So here's a quick screen capture of P6MC 2.0. So you can see the phantom here, and you can see the field size regenerate, as well as the X-ray energy and the MAS involved. So there have been a couple studies comparing P6MC to commercially available phantoms. So in particular, Ma et al. Compared the planar radiography dosimetry of the newborn atom phantom with P6MC, and they found quite a large variability in the organ doses from -41% to positive 87.5%. So for cone beam CT, Wood et al. In 2015 compared P6MC simulation of prostate CBCT with the rando phantom. And again, they found quite large differences of 80 to 60% differences in the liver and stomach. However, this was attributed to the edge of the field where the X-ray beam was different between the two modalities. So this raises some uncertainty in P6MC's adaptation for cone beam CT, because remember P6MC is for planar, but it can be adapted for rotational scripting as well. And for slot scanning, PI et al. In 2023 assessed P6MC simulation for slot scanning radiography, and they found up to -95% and positive 67% dose differences in organs. So they concluded that P6MC is actually not suitable for slot scanning. Which brings me to the problem and the significance of this study. So quite often a lot of modern complex radiology techniques exceed the abilities of standard simulation software like P6MC. So this facilitates the need for more physical phantoms. Quite often the problem is though that commercial phantoms are prohibitively expensive, costing upwards of $40,000, and they're not easily customizable. So for example, the Atom Phantom family series here are one of those commercially available phantoms. So especially in pediatric radiology, where we need lots of different phantoms to cover a range of patient sizes, this becomes an increasing problem. And additionally, the existing phantoms like the atom and rando phantoms differ in both composition and design from the standard phantoms used in those simulations, so like the Christie phantom and the MIRD phantoms. Which brings me to the aims of this project. So this project was aimed to develop and validate a cost-effective pediatric dosimetry phantom based on the newborn model by Christie and Eckermann using 3D printing. So this way it allows our phantom to be directly comparable to P6MC, as well as CT Expo and Radomatrix. So this was achieved through three main objectives. So objective one was to investigate 3D printed filaments with suitable attenuation properties across a broad range of diagnostic energies. The second aim was to design and construct the pediatric dosimetry phantom based on our findings from aim one. And then thirdly, after the phantom was completely constructed, we wanted to validate the dosimetry outcomes against P6MC 2.0 Monte Carlo simulation. So let's go straight into aim one. So tissue equivalency of 3D printed materials. So there were a couple of candidate materials that were recommended by the literature, and we printed them using the Bambu Lab X1 carbon 3D printer at various rectilinear infill densities, and they underwent a Hounsfield unit analysis. So the first one was polylactic acid, or PLA, which is one of the most commonly used 3D printed filaments. And this was used to simulate soft tissue and lung tissue. So we printed it from 100% infill density down to 15% infill density, as you can see here, 100% down to 15%. And then for bone replication, we printed stone-filled composite PLA by Form Futura. So stone-filled composite PLA is actually PLA filament filled with 50% ground stone powder, which does two things. It increases both the density of the material as well as increasing the effective Z number. So a lot of the literature surrounding 3D printed filaments and tissue equivalency suggested stone fill was a good alternative for bone. So we printed these from 100% down to 55%. So for Hounsfield unit analysis, we conducted them over a wide range of diagnostic X-ray beam qualities. So for CT beam qualities, we used the Siemens Somatom Force from Perth Children's Hospital, and we scanned our samples from 70 to 140 kVp, a half value layer of 4.8 mil to 8.6 millimeters of aluminium. For radiography, so this is a unique part of our study, is that we were able to conduct Hounsfield unit analysis at beam qualities typical for radiography. And this was done using the Bruker SkyScan 1176 preclinical micro CT at the National Imaging Facility at Harry Perkins Institute. So this was done for 40 to 70 kVp X-rays with a half value layer of 1.3 millimeters of aluminium to 2.2 millimeters of aluminium. So let's go straight into the results of our findings. So HU results for newborn soft tissue. So you can see here are the ROIs we generated. They're a bit offset actually here, but from these ROIs, we measured the average Hounsfield values, as you can see here, across the different infill densities. So for clinical CT, we can see that we've got a very strong correlation coefficient between the Hounsfield units with the infill density. Same thing with the micro CT, except we have less data points. So the attenuation of PLA demonstrates a strong linear trend in both CT modalities. And this was consistent with findings from other studies like COSY and MAR. So now if we plotted them against the beam quality, so in this case, the Hounsfield units against the half value layer, we can see that soft tissue, the theoretical soft tissue was calculated as well, exhibits almost no energy dependence at all. And that's expected because soft tissue is mostly water equivalent. So we won't see any difference in HU with increasing beam quality. However, we can see that HU of PLA is increasing with beam quality. And that's because the effective Z number of PLA is thought to be less than the effective Z number of water, being 7.25 compared to 7.42. So this means that at lower energies, the photoelectric absorption of PLA is less than the photoelectric absorption of water, decreasing the HU of the PLA. But overall, we can still see it's quite a similar trend to soft tissue. We've just zoomed in. So next, we will talk about the results of our newborn bone, so our theoretical newborn bone versus our stone fill. So we can see here again, Hounsfield units, except now from 600 to 2000, and our half-value layer. So this is micro CT and clinical CT once again. So you can see that the HU of the stone fill shows a stronger energy dependence than the theoretical bone. And this was again thought to be because of the difference in the effective atomic number. So the effective atomic number of stone fill is thought to be higher than the effective atomic number of newborn bone. And the reason why is because newborn bone is got a higher oxygen content rather than calcium content because of the higher amount of red bone marrow. So again, we see an increased photoelectric absorption, but we also see similar trends in the literature for our stone fill trends with energy. And then finally, our HU results for lung. So low infill densities were printed and scanned at clinical CT energies. And the reason why is because we can see that there's almost no energy dependence. It's very, very minor. So there was no point scanning at the micro CT level because we knew that the energy dependence was pretty linear anyways, pretty flat. So now onto final filament selection. So we calculated the optimal infill densities at each of the 15 beam qualities using a linear regression model at each of the beam energies. So this gave us a list of optimal infill densities for each tissue type. So for example, if we look back at figure 9, so we can see that the HU response to PLA with infill density of at 100 kVp. So the theoretical attenuation of soft tissue is about 32 HU. So you can see that correlates to about a 91% PLA infill density at about 100 kVp. So this allowed us to generate a list of 15 infill densities across the diagnostic energy range. And so the overall optimal infill density was weighted by the relative frequencies of each X-ray energy used in pediatric radiology. So for radiography, we can see that most of the energies are around 65 and 50 kVp, whereas for CT, most energies are around 70 or 100 kVp for pediatric radiology. So our biggest weighting would have been in this area with 65 kVp and 70 kVp. So this arrived at a newborn soft tissue equivalency of 93% white PLA. For bone, it was 81% stone fill. And then for lung, it was 26% white PLA. So we can see here, we've plotted the theoretical attenuation of bone, soft tissue, and lung tissue alongside the attenuation of about 81%, 93%, and 26% samples. So you can see that across the diagnostic energy range, especially for lung tissue and soft tissue, our samples exhibit a very strong suitable attenuation properties. So now onto aim 2, which was the computational phantom design, or the design and construction of the phantom. So Rick and I generated STL files in MATLAB for the tissue regions of the Christian Eckermann phantom. And so you can see here's a GIF of the phantom in axial slices. So the phantom included 40 organs, suborgans, and bones, and we split it into 21 slabs, 25 millimeters thick, kind of like the rando and atom phantom. So altogether, we fitted 186 TLD holes. Each hole was about 5.2 millimeters in diameter, which gave us on average, about 4 TLDs per organ. 56 additional holes were also added to the phantom and to be filled with alignment rods. So using the infill densities presented before, so 93%, 81%, and 26%, we printed the phantom's 21 sections. And we can see here with all the TLD holes as well as the organs have been visualized in red. So red wasn't used throughout the entire slab, just on the surface, just to visualize where the organ was. So you can see here, high density PLA for soft tissue regions, low density PLA for the lung tissue regions, as well as the high density stone fill for the bone regions. So these were printed on the Bambu Lab X1 Carbon with automatic material system and gave us a total printing time of about 135 hours with a total phantom mass of 3.6 kilos and a total material cost, including reprints, of about $330. So the total height of the phantom was 51 centimeters as well. So here's me putting the phantom together in this frame. So you can see here the phantom measured about 51 centimeters in height, not including the frame. And then under an X-ray image, the phantom exhibited suitable attenuation characteristics. So you can see the bony regions exhibited higher attenuation compared to the soft tissue, which also exhibited higher attenuation compared to the lung tissue. So this is just a little time lapse of me assembling the phantom in this frame. Cool. So now we're on to the exposure portion of our project. So this project used lithium fluoride, magnesium, titanium low-dose thermoluminescent detectors from Royal Perth Hospital. And so prior to exposing the phantom, we actually calibrated the TLDs in terms of air coma. So we used the RaySafe X2 RF diode dosimeter for this, and we exposed the TLDs with the same beam qualities as the phantom. So altogether, we had 6 batches of 50 TLDs, where from each batch, we needed 12 calibration rods. And out of those 12 calibration rods, it was 3 TLDs at 4 different dose points. So for example, for our exposure, we used 0, 1, 10, and 20 milligray for our dose points. And these were used to generate a linear calibration curve. So here's just an image of us calibrating the TLDs. We can see one here was the 1 milligray TLDs and the 10 and the 20. So these are from 3 different batches. And so each calibration batch had a strong linear regression of R squared being greater than 0.999. So finally, we're onto our phantom exposure. So the phantom's holes were filled with TLDs, as well as the TLDs were also positioned on the surface for skin dose measurements. So here's an image of the phantom on the digital diagnosed C90. We exposed it to a 60 kVp X-ray with 3.5 millimeters of aluminium filtration and 32 milliamp seconds exposure. So we exposed the phantom 20 consecutive times. You can see here that the phantom is not actually in its frame because the frame wasn't built just yet. It was still held together using tape. But you can also see the TLDs on the surface of the phantom here. The RaySafe diode dosimeter was also placed next to the phantom for a simultaneous measurement of the X-ray beam. And then this red arrow here indicates the cathode to anode direction. So it's pretty important to know for later, but the anode was on the left side, on the head side. So we exposed the phantom with a source to table distance of 102 centimeters and a 50 by 50 field size at 110 centimeter SID, which was almost equivalent to a whole body exposure. You can see that the legs are slightly cut off. And so for the phantom dosimetry, as I mentioned before, the TLDs were calibrated in terms of air coma. So they needed to be converted to tissue absorbed doses. So we used the method inspired by the AAPM TG61 protocol for kilovoltage X-ray dosimetry, which allowed us to calculate the air coma to water through the mass energy absorption coefficients, MEACs for short. And then we can also assume that the dose to water is equivalent to the air coma, and that's because our energy is so low. So it's essentially this equation here, D water is approximately equal to K water, which is equal to K air times the ratio of the mass energy absorption coefficients. And then finally, we could calculate the tissue doses using the MEAC ratios of tissue to water. So here's a table of our specialized ratios. So water to air for our specific beam was 1.017. And then for soft tissue to water as well as bone, these were all P6MC specific compositions. So we calculated these using the NIST 126 data. So now onto validation with P6MC 2.0. So we simulated the exact same procedure in P6MC. So 60 kVp, 50 by 50 field size, almost a full body exposure and everything. Same filtration, same tube anode angle. And we can see that our phantom exhibits pretty close dosimetry qualities to the simulation. So for example, our clavicles here exhibit quite a good conformality, but some organs like the ribs show a bit more of a difference, and some organs like the pelvis also show a bit more of a difference. So here we have organ absorbed doses and our organs here, and physical is in blue and simulated is in orange. So taking a closer look, here is a plot of the percentage differences from negative 40% to positive 60%. And now I've actually rearranged the organs from superior to inferior. So we can see here that the organs in the superior region actually had underestimated a dose in our phantom compared to the simulation, whereas our phantom overestimated the dose in the inferior regions, except for in the bone, in the lower leg bones. So it was mostly in the lower trunk, like slabs 0 to 4. So this was thought to be caused by the anode heel effect, because if you remember from the image before, the anode was on the head side of the phantom, which meant that the head side would receive a harder beam, which would reduce the dose, whereas the cathode side would have a softer beam, which would likely increase the dose. So the reason why this is different is because P6MC doesn't actually consider the tube orientation. It only considers the tube anode angle for generating the spectrum, and not actually the different spectrum over the field size. But still, even with that, 40 out of our 45 organs are within a 25% dose limit of the simulation, with the average relative difference only being 5% higher. So this is still a much better performance than any other phantoms seen in the literature when compared directly to P6MC. So now this goes to brings me to my discussion, which is where the anode heel effect, which I already discussed, is a point of uncertainty. Second point of uncertainty is the discrete positioning of the TLDs in our organs. So P6MC has a geometric volume where the dose is averaged over the entire organ volume, whereas our dose measurements have some kind of sampling uncertainty because we have a discrete number of positions. So for example, in our liver slice here, we can see that we've got a TLD near the anterior side, in the central, and in the posterior side. So this could possibly be a reason why our phantom overestimated the dose, is because this TLD here would have received a higher dose as it was closer to the source. As well as the intrinsic uncertainties of lithium fluoride TLDs as well. So Sedek et al. Claimed that TLDs, or lithium fluoride TLDs, have a plus or minus 10% uncertainty with a minimum measurable dose of 0.5 milligray. But this would have been minimized by the fact of us using lots of TLDs in each organ. Additionally, the linear attenuation of the selected PLA and stone-fill filaments were generalized over the whole diagnostic energy range. So it wasn't specifically set for 60 kVp, which could also be another reason why we have a different attenuation compared to the P6MC simulation. Which brings me now onto my limitations. So standard PLA may have unknown additives. So even though we selected standard PLA, like generic PLA from Bilby 3D, every manufacturer has their unknown additives for either color or stability of the filament. So this actually may affect the experimental reproducibility, especially if we're going to use other brands of PLA. Also, the consistency of the stone fill between batches is questionable. And this is also, again, highly dependent on the manufacturer, because Form Futura create their own stone fill PLA, which means that they might have added different amounts of stone powder between each batch. The phantom is also highly dependent on the printer's quality. So for example, if the printer is not serviced properly, or if the print settings are wrong, then we can also decrease the quality of our print, which decreases the phantom's quality. Another thing is PLA is biodegradable and may deform over long time periods, but our study hasn't seen anything yet, so that's good. And then again, similar to what I mentioned before, dosimeters have their intrinsic uncertainties, like the TLDs and the RaySafe have their own uncertainties, as well as the dose simulation software uncertainties, which I mentioned about P6MC. So now onto future work. So we want to further construct our phantom for different phantom sizes. So for example, this image here is the different phantom sizes used in P6MC. So we want to construct different sizes, as well as possibly implementing GAF chromic film for KV dosimetry measurements. And then we want to compare our dosimetry of our phantom to CT Expert and Radiometrics, so other dose simulation software, as well as expanding to other imaging modalities, such as cone beam CT, DXA, and CT. And then maybe also being able to manufacture our own composite filament for bone. So in conclusion, this project was successful in achieving its aim of developing and validating a 3D printed newborn dosimetry phantom for pediatric radiology. So aim 1 was to investigate 3D printed filaments with suitable attenuation properties across diagnostic energy range, which we were successful in doing. We found 93% PLA for soft tissue, 81% stone fill for bone tissue, and 26% for lung tissue. Aim 2 was to successfully design and construct the pediatric dosimetry phantom, which we were successful in doing again. Then aim 3 was to validate the dosimetry outcomes of our phantom against P6MC 2.0 Monte Carlo simulation results, which again, our results were quite close conforming to the simulation and actually better than anything seen in the literature so far. So finally, just to finish off, a couple of acknowledgements. So I'd like to thank my supervisors, Rick and Pejman, for your guidance throughout this project. Couldn't have done it without you guys. The Medical Technology and Physics Department at Sir Charles Gairdner Hospital for purchasing the materials used in this project, as well as the MTP staff, so Mario, Sinead, for helping me in my research and my work experience. Perth Children's Hospital, all the radiology staff at Perth Children's Hospital for helping me use the CT scanner and the radiography equipment, as well as Royal Perth Hospital, so Marsha and Chris for the clinical physics department for providing the thermoluminescent detectors, as well as the National Imaging Facility at Harry Perkins. So thank you, Diana, for showing me how to use the micro CT. And then UWA for providing the 3D printers used throughout this project and financing the micro CT measurements, as well as Adriano for helping me quantify my uncertainties. Thanks for your attention.
Thanks very much, John. Great presentation. We'll open up for questions now. If anyone has any questions.
I have just one quick question. You can test the effect of Donald Hill if you just, do your measurements and repeat the measurements, the phantom rotated 180 degrees, like head first, feet first configuration.
Yeah. Yeah. Awesome.
Any other questions for John?
I have a question.
Okay. Go for it, Andrew.
I was just wondering if you kept the volumetric flow the same throughout all the prints, or all the printing parameters, and if you changed that, or if you had recommendations for someone wanting to implement your phantom or make your phantom, if you would have any recommendations for them.
Yeah, so the volumetric flow rate that we used was the standard 20 millimeters cubed per second. And then for like printing parameters, I found that using a hotter bed temp, so 60 degrees, was actually better because it reduced any warping. That way the print stayed flatter for longer. But I mean, if I go back to this, we can see the density of the PLA and the density of the stone fill and the lung tissue. So if someone was able
That's a really good point.
Exactly.
Cool. Good work.
Thanks, Andrew.
Roger, you have a question?
John, that was a highly professional and very enjoyable talk. Thank you very much. Very quick question. Conceivably, although it might have a very narrow application, but I'm thinking about a problem that I was thinking about the other day in respect of DEXA. You could measure or predict, both predict and something while the child is being scanned. You could, in theory, do that, couldn't you?
Possibly, because our stone fill will have maybe we need to fine tune our stone fill some more, but it should have the same attenuation characteristics and scatter characteristics as an actual pediatric patient.
Yep. Okay, thank you.
Does that answer your question?
Yes, it does. Thank you.
Okay, thank you.
Thanks, Roger. John, just while we're on this slide, how did you calculate the, is that 95% confidence intervals or uncertainty?
Prediction interval. So yeah, it was the prediction intervals. That was, so I think, I don't remember the exact equation. Adriana recommended it to me, but it was using the mean square error. So for example, the 81% stone fill. So the mean square error between this blue line and the purple line, and then using the square root of 1 over n, so some kind of specialized equation. But it involved the mean square error.
Gotcha. Yeah, cool. And on this last question, I was just wondering if there was any data on the stability of the stone fill between manufacturer batches in the literature, which you mentioned what might be a limitation of the work.
Yeah, I haven't seen anything so far because in the other studies, they all exhibited very similar attenuation characteristics to ours, except for the new batch of stone fill, which I tested later on, and it exhibited a lower linear attenuation compared to the old batch. So I haven't seen anything so far, but that's something we need to investigate further.
Yeah, cool. Thanks, John. We'll go for the last question with Godfrey.
Hi, John. Great presentation and nice. You did a great work. Thank you. I just wanted to ask about how you characterize the properties of the PLA or the 3D printing material. You were using the HU numbers. How good is that to characterize the properties of the radiation properties of the material? Do you think would you have done it maybe differently or another method that you could have used?
So, sorry, I don't think so what do you exactly mean by radiation properties? Do you mean like linear attenuation?
Yes, so you characterize this, you are looking at the properties of that material so that you can say they are equivalent to the, maybe you are trying to find the equivalent to the bone, equivalent to soft tissue for the pediatrics. So the question is that, do you think that the using HU as a way of validating the properties of that material is good enough?
So yeah, you could definitely take it further, but all other studies in the literature do it in terms of HU or linear attenuation coefficient, but you can take it further and probably go into electron density and the effective atomic number as well.
Okay, so what do you think could be the limitation of just using the HU numbers?
Well, I guess the limitation is the fact that it doesn't perfectly tell you the scattering and mass attenuation coefficient characteristics. So, especially in high energy dosimetry where charge particle equilibrium is important, I think that's a limitation.
Okay.
Excellent. Thanks, John. Thanks for all the questions. That's all the time we have for questions, so we'll finish it there. Again, great presentation, John. Very well done.
Cheers.